Okay, so I'd like to talk a little bit about bacterial growth, the way that bacteria grow, the dynamics of its growth, and sort of the requirements for its growth for different types of bacteria. So, first off, most bacteria grow by dividing through binary fission. And that just simply means that the cell splits in half, but it's a little bit of a more complicated procedure than that. The first thing that happens is that the DNA, as you can see up here at the top of this image, is going to replicate. All right, so the chromosome divides, and different parts of the chromosome of the two new separate chromosomes are going to attach to the cell membrane, and they will migrate to opposite sides of the cell. Then there's a ring that forms around the center of the bacterial cell, and this ring is essentially like a belt. You know how with a belt you can pull it tighter, but it has those little prongs in it, and that stops it from getting looser? That's the way this belt works. It can tighten, but it can't loosen. So this belt gets tighter and tighter and tighter and tighter, and it's hooked to the membrane around the center of the cell. And so as it gets tighter and tighter and tighter, it's going to pull the cell in bit by bit by bit to this ring that gets smaller and smaller and smaller. And you have to think about the cytoplasmic membrane as being sort of similar to a soap bubble, like one of those little soap bubbly, blowy things that you get at birthday parties when you're a kid. And if you've ever seen two of those soap bubbles kind of collide in the air, um, they essentially sort of like they touch each other, and then they go bloop, and you get one big soap bubble. This is essentially that same process, but in reverse. You're teasing the cells apart and apart and apart, and you're making the area between them thinner and thinner and thinner until it eventually becomes sort of more stable for it to split into two different soap bubbles, or in this case, cells. And when that happens, the cell is going to, it's going to be two cells, but it's temporarily going to be two cells inside of one cell wall. It hasn't quite finished splitting yet, but it's very quickly going to build a cell wall down the middle of the cell, and then, boom, you've got two cells. Now, this takes different types of cells dramatically different times. This is the time of division. It's sometimes called the doubling time or the generation time. And in most cells that you work with in a laboratory situation, this is going to be fairly quick. So for instance, for E. coli, under ideal conditions, this takes about 20 minutes. That's with perfect conditions. Like it's got absolutely great nutrients. It's got exactly the right amount of oxygen. It's at its favorite temperature. It can take about 20 minutes. Under more typical out in the world conditions, could take more like an hour. Even still, that's pretty fast. And that's a doubling time. So it starts off slow, but then it goes into what we call exponential growth. So basically you get 1, you get 2, 4, then 8, then 16, 32, 64, 128, 256, 512, 1024, and it just goes on and on and on. And it gets really big really fast. Other organisms take longer to divide. For instance, Mycobacteria tuberculosis, which is the bacteria that causes tuberculosis, has a doubling time of about 24 hours, about a day. And so it's going to have that same exponential growth cycle, but it's going to be a much more elongated one because the doubling time is longer. So this is the equation that is used to calculate the number of cells in a population. And it's basically the total number of cells, NT, is equal to the initial number of cells, which is N0, times 2 to the number of generation cycles that's been around. So, if we're talking, for instance, E. coli, and it's been there for 1 hour, okay? And you put in one cell at the beginning, and it's been in there for 1 hour, it's going to be 1 times 2 to the third, because there's 3 20-minute periods in 1 hour. 2 to the third is 8, so you're going to have 8 cells after an hour. Pretty easy. Now, I recall when I was a child, my mom sort of explained exponential growth to me in this way. She said, would you rather have $1 million, like just right now, today, that's it? Or would you rather have a penny today, and then tomorrow I give you 2 pennies, and then the day after I give you 4, and the day after I give you 8, and we keep that up for 2 months, and then we're done? Okay? Well, which do you think? And of course, the obvious answer is you should take the $1 million, so that's the wrong one. You end up with a lot more money if you go for the penny. And for the first week, you're looking at the guy who's got his $1 million, and you're thinking, oh man, I got gypped. But pretty soon after that, it's going to catch up, and within 2 months, you're actually going to end up with a whole lot of money, with a whole ton of money. Let me take a look here. Just a second, I'm trying to... 1 second. Okay, so according to my calculations, after 60 days, 2 months of this like penny doubling every day thing, you would end up with 1.15 times 10 to the 16th dollars. Now, times 10 to the 9th is a billion. Times 10 to the 12th is a trillion. So we're looking at something in the nature of like 10 to 100 quadrillion dollars. That's more money than exists in the United States, probably. I mean, you can figure our national debt is somewhere in the realm of 1 to 2 trillion, and this is 1,000 times that. So it's definitely a little bit of a better deal than getting $1 million right away. And the point of this anecdote is to explain how exponential growth works in bacterial cells. Okay? So we established that if you start with 1 bacterial cell, 1 E. coli, then after an hour you're going to have 8. Okay, that's a bit of a jump, but it's not exactly huge or unimaginable. Well, what about if you instead wait 4 hours? 4 hours is a decent amount of time. That's going to be 1 times 2 to the, 3 times 4 is 12, so 12th power. And that is equal to 4,096, as you can see down here. And so after 4 hours, you're going to have about 4,000 cells. Again, 4 hours, 4,000 cells, that's not bad. They're small. After 4 hours, you might not even be able to see them. Maybe it's gotten a little bit cloudy or something like that, but that's not undoable. What about after a whole working day? 8 hours. So that's going to be 24 growth periods. Well, 2 to the 24th is, now we're talking 16 million. 16 million is a lot of cells. It's not an unimaginable amount of cells. That's certainly a number of cells that you can deal with, but that's a lot of cells since you started with one. You came into work in the morning, you started with one cell. When you leave work that night, you've got 16 million of them. That's a big jump. What about the next day? You let them keep growing and you come in the next day. It's been a full 24 hours. Well, at that point, so it's going to be 24 times 3, that's 72, and 2 to the 72nd power, you'd have about 5 times 10 to the 21st cells. So if a trillion is 10 to the 12th, we're talking about like a billion trillion cells. So you're thinking a trillion cells? Yeah, you've got a billion times a trillion cells. That's a lot of cells. That's probably more cells than you could really reasonably work with. That's like a room full of cells. If you let it go on for a week, and bacteria could actually grow until, without any sort of limits, that they could continue replicating every 20 minutes, within a week, you would have more bacteria than the entire mass of the planet. Obviously, this doesn't actually happen. And that's because there are limits on bacterial growth, and we're going to talk about some of those limits on bacterial growth in just a second. Last thing that I want to point out here is that, you remember we said that Mycobacteria tuberculosis has a doubling time of 1 day, of 24 hours. So in that 1 day, you got a billion trillion bacterial cells, if we're talking E. coli. In that same period, you've gone from having 1 tuberculosis cell to 2. So that generation time can make a lot of difference. 1 to a million or a billion trillion versus 1 to 2. This is one reason why they usually don't test for tuberculosis by actually swabbing and trying to grow a culture of it. If it's only dividing once every day, then it's going to take at least a month to grow a reasonable amount of cells that you could actually test. After a month of growing tuberculosis, it's about roughly equivalent to growing E. coli for maybe 4 hours. That makes it a kind of hard organism to work with. There are people who do so. My hat's off to them. So here we just see a blow up of the process of cell division. Okay, now, I said that there are natural limits to the way that cells can grow. Cells can't grow without limit forever. And so there is a particular mode of life that cells enter into. And this is called the growth curve, and it's usually typical of cells that are being grown in a laboratory environment. So we're talking about cells under ideal situations. Now, to explain basically what's going on here, I want you to think about what's going on from the point of view of the cell. Okay? So it's going to start off, it's in storage. It's cold, it's dark, there's very little to eat. Suddenly there's a bright light. A door swings open, and a man or a woman, let's not be sexist, a person in a white coat grabs you and transports you to what can only be described in bacterial terms as paradise. This is the promised land for bacteria. It's exactly the right temperature. It's got all of your favorite things to eat. It's got endless, boundless fields of room. And it's got, the exact right amount of oxygen. And it's just absolutely perfect. For a bacteria, this is like the best time of its life. And I can tell you, as a somewhat large person myself, that one does not simply look at a banquet like that and just go right into it. You have to kind of work up to it. You have to sort of sharpen your knife and fork, tuck your bib in, take a good deep breath, savor the moment. And bacteria do somewhat the same thing. They are going to take a little bit of time right after being introduced into this situation where they will prepare. This is called the lag phase, and you can see it here at the beginning of this curve. And what happens in the lag phase is that the bacteria is going to change its genetic expression. And it's going to begin expressing the genes that are associated with eating lots of stuff and dividing, reproducing. This takes, well, it depends on the bacteria, but it's going to take between 10 and 15 minutes usually. And then once this preparatory lag phase is done, it enters into the second stage here. And that second stage is called the log phase, or the logarithmic phase, or the exponential growth phase. During this phase, the bacteria is basically doing two things. It eats and 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 eats, and then it divides. And then it eats and 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 eats, and then it divides. And that's basically the only things that it's doing. It's got so much food and so much space that it just keeps eating and then dividing and then eating and then dividing and then eating and then dividing. This is where it's growing according to that equation we were just looking at. This is where it's doubling every 20 minutes, or whatever its generation time is. There's a problem. No matter how rich or glorious this paradise that it's been transported into is, well, when you start abusing that kind of privilege, it eventually runs short. And even though this thing started off with like so much to eat, within a few hours, maybe a day at most, it's about as long as the log phase can last. Within that time, it's going to start to look kind of unpleasant. All of the good stuff has been eaten by now, and even though it looked like it was very roomy at the beginning when there was just one of you, now that there's, a billion trillion of you, it's getting kind of crowded. And you've eaten up all of the good stuff to eat, all of that initial supply of food. But even though all of that initial supply of food has been eaten, there is still one food source left. You spot it? That's right. You can eat each other. That's when you enter into the stationary phase. Look at the top of the curve here and see that stationary phase. During stationary phase, birth and death are equal. That's because there's only so much food to go around, and most of that food is already contained in bacteria. New bacteria are still being born, but in order for there to be enough energy for one new bacteria to be born, that means that one old bacteria has to die to liberate those nutrients. So there's another genetic changeover that happens during this phase. The bacteria start expressing, they kind of tone down their reproductive genes, but they really, really ramp up their scavenging genes. Because now it isn't their favorite food that's around here. Now what's around here is stuff that they can eat, but it takes a lot more work for them to gather. And they're competing against a whole bunch of other bacteria to see who's the best at gathering nutrients. And that's the guy who's going to manage to scavenge enough of them to reproduce. This stationary phase can last for quite a long time. Well, relatively speaking. It can last for a lot longer than the log phase because you're not actually getting any increase in the population. But it can't last forever. It can usually last, again, depending upon the species and the size of the container that they're in, days to a week, somewhere in that range. More than a day, less than a week. But even it doesn't last forever because even if all of the bacteria are just eating each other, you can't keep recycling things infinitely. There's this little problem we have, and it's called the second law of thermodynamics. And what the second law of thermodynamics states is that, well, basically what it says is that entropy always increases. What that means is that anytime energy is used, it doesn't disappear. It doesn't go away. Energy can't be destroyed. That's the first law of thermodynamics. So the energy doesn't just disappear, but what happens when you use energy is you change it. You change it from one type of energy to another type of energy. And it could be changed back. But every time the energy is changed, some of that energy is lost. And again, it doesn't disappear. It doesn't go away from the universe. What that means is that anytime you change one form of useful energy into another form of useful energy, like you're changing, chemical energy in the form of glucose into lipids to make your membrane, or into the motion of a flagella that's going to move your cell around. You're changing one type of energy into another. Those processes always involve some of that energy being changed into a less useful form. That's called waste energy. Often it's heat. Sometimes it's chemical byproducts that can't be reused. Sometimes it's, it can be lots of other things as well. But mostly we think of it as being heat. And that heat energy, or that waste energy that gets lost, it can no longer be used by the bacteria. So sort of over time, even if you're recycling and you're eating mom and dad, and your kids are eating you, and then their kids are eating them. So this is kind of this horrible Mad Max apocalyptic wasteland style thing. Even then, every time that happens, some of that energy gets lost as waste. That means that over time, eventually, there will not be enough energy in the system to support the system's population. This is when we enter into the death phase. During the death phase, it's a slow, long phase. So the bacteria ramped up in a very exponential fashion. Like you went from 0 to 1 billion trillion in like a day. The death phase is going to take a lot longer than that to go down, because you're going down in a linear fashion. You lose about the same amount of bacteria every day that you can't get back. During the death phase, bacteria aren't really trying to reproduce anymore. This is really nothing, there's no nutrients to really allow them to. Some of them are going to really ramp up their scavenging genes and try to scavenge as much energy as possible. Some of them, probably the smarter ones, are going to shut down. They're going to turn off their metabolism to the best extent that they can. They're going to not make any new components, not grow at all. They're not going to try to move. They're not even going to make new proteins, not even if those new proteins might help them find some food, because there's just no return on the investment. If you're running around 10 miles a day and all you find is like 5 berries, Well, those berries don't give you enough calories to compensate you for the amount of work you had to do to get them. So at this point, the best strategy is really to just shut down and go into suspended animation. And that's what a lot of the bacteria do. So the ones that remain active, they kind of die off at a steady rate, and more of the bacteria go into this sort of, shutdown mode. And once enough of them have gotten into the shutdown mode, and this death phase can last for months, right? Because you've got, you may have 1,000 cells dying off per day, but if you've got 1 billion trillion cells, it's going to take a long time for them all to die out. Those death phase can last for a long time, and then it sort of eases into this sort of fifth stage, which may be a stage, or maybe just sort of an extension of the death stage. It depends on who you ask and what book you look in, but this phase of prolonged decline. This is once all of the stupid cells that are trying to remain active, they're dead. And at this point, all you really have left are mostly the ones that are in suspended animation. And they can stay in that state for years. I know in my research, I've occasionally gone to the fridge and retrieved a bacterial culture that I stuck in there like 2.5 years ago, and I was able to get it to come back and regrow them. In all likelihood, 99.9999% of them were dead. But even that 0.001%, when you're talking about an initial value that was so high, that was a billion trillion cells, well, 0.00001% of that is still like 1,000,000 cells. So there's still enough living ones in there that you can get them back. It's going to take them a long time to completely die out. That's the bacterial life cycle. We've gone from the beginning all the way to the end. We started off with a single cell that came from a hibernatory environment. We put it into paradise. It did its little preparatory thing, and then boom, off to the races, and it's growing like mad. And then things really do go mad because there's not enough food left, and it's, kill or be killed, and lots of bacterial cannibalism, and then the slow, long decline of death, and back to where we started in suspended animation. That's pretty much what the bacterial life cycle is like. And it's, it's fairly typical, not just of bacteria, but really of a lot of things. Now, what we were just talking about was the bacterial life cycle in an idealized environment, like when you're growing them in a lab. But out in the real world, there are a bunch of other things that are going to come in and screw around with that curve. Because out in the real world, the bacteria aren't being dropped into paradise where everything is perfect. Out in the real world, bacteria are being dropped into, like New Jersey. Right? New Jersey's not bad, but I wouldn't call it paradise. It's okay, but there's, a lot of stuff that could be better. So I want to talk about some of these environmental things that are going to affect the way bacteria grow. They break down into basically 4 big categories: temperature, oxygen, pH, and water availability. We're going to tackle them in that sequence. So in terms of temperature, you have basically 5 different classifications of temperatures. And you have bacteria that will live in each of them. The first, on the left of this graph, are the psychrophiles. Psychro meaning cold, phile meaning to love. They love it cold. They're like that crazy friend of yours that goes skinny dipping in ice water. Here in Arizona, where I am, that's not, there's not a whole lot of ice water around. But these guys are kind of crazy cold lovers. And they live from -5 degrees Celsius, that's below freezing, to about 15 degrees Celsius, which I think of as being a chilly autumn day in the Northeast. It's long sleeves and jacket weather, but it's not like put on your parka. This is when you exhale and you can sort of see your breath, but it's not like snowing at 15 degrees. That would be snowing at, 0. So these psychrophiles, from a human health perspective, are not super important. You're warm, they like it cold. So they're not going to live inside of you. Most of them make very poor pathogens. Some of them are involved in food spoilage in like the refrigerator, because the refrigerator is between this, -5 to 15 degrees Celsius sort of range. But even then, these are mostly going to be bacteria that you find in like the Arctic and the Antarctic regions, or the frozen tundra, really cold places, places that are cold and that stay cold. Okay, one step up from that is psychrotrophs. Psychro means cold, as we said. Troph means to grow. They don't love it cold, but they grow in the coldish type of regions. And even though it says cold as in psychro, it's not actually all that cold. These are centered from 20 degrees Celsius to 30 degrees Celsius, and room temperature is generally considered to be 25. So these are the guys that live in, like, out there in the normal environment. Normal room temperature environments. That's about 72 degrees Fahrenheit, or 25 Celsius, as we said. So I think of 20 degrees Celsius, just to get you an idea of sort of what these mean. I think of 20 degrees Celsius as the first day of spring that you think about putting your jacket on, and then you go, no, it's a nice day. I'm just going to walk outside without my jacket. And it's still a little bit chilly. It's colder than you like keeping your house, but it's a nice day. That's 20 degrees Celsius. 25, as I said, is room temperature, so look around your room. That's the temperature. 30 degrees Celsius, this is, hotter than most people keep their room, but, if it's a hot, Phoenix day, and you don't want to drive up your AC bill, that's the temperature that you keep your house. It's like around 85 degrees Fahrenheit. I'm not calculating this exactly, but it's definitely hot. But it's not, it won't kill you. It's less than 100 degrees Fahrenheit. It's less than going outside in the desert, but it's uncomfortably warm. Psychrotrophs are mostly, environmentally, they're very relevant, because most of the stuff that just lives out there in the environment, in the soil, tend to be psychrotrophs. They're living at room temperature. Room temperature is, the standard temperature. So most microbes that exist are probably psychrotrophs. In terms of human health, they're they're more relevant than psychrophiles, but less relevant than some other stuff. They're often involved in food spoilage, because if you think these are the microbes that live at room temperature, if you take like your, mayonnaise and you leave it out overnight on your counter, what's going to be growing in it? Well, stuff that grows at room temperature. And up until the last 100 years, pretty much all of our food was kept at room temperature most of the time, because we didn't really have fridges. So these are important in food spoilage, and in some cases of like food poisoning or food sickness. Okay, the next bracket up is mesophiles. These go from 25 degrees, that's room temperature, to 45 degrees. Bracketing 37 degrees. 37 degrees is body temperature. So these guys are right around body temperature, and most pathogens fall into this category. Most pathogens are mesophiles. Meso meaning middle, phile meaning to love. They love living at sort of middle temperatures. So most diseases fall into this category, as do most of the natural flora that lives within you, most of the natural bacteria that lives in you all the time. So just to describe these brackets, 25 degrees is room temperature, 37 is body temperature, 45 degrees I like to think of as you've drawn yourself a nice hot bath. And like you put your finger in, you go, ooh, that's a nice hot bath. And you don't crawl in all at once, right? So you got to kind of work into it. It's not going to hurt you. It's not scalding, but it's a nice hot bath. That's about 45 degrees Celsius. Thermophiles. Thermo, heat, phile, to love. These like it hot. Some like it hot, right? So these go from like 45 degrees, that's that nice hot bath, to 70 degrees. 70 degrees Celsius is scalding. This is kind of like a low simmer. All right? It's not boiling, but it is water that if you dip your hand in it, it's going to burn. These are bacteria that you find in hot springs. And when I say hot springs, well, yeah, so 45 degrees is going to be the low end of a hot spring. That's the type that, like an onsen or something that you would find in Iceland or something, one of those places where you could bathe. 70 degrees is going to be more like the sort of hot spring that you find in, Yellowstone. And there might be a runoff from a geyser that comes into it or something. It's hot. Don't touch it. They sometimes have all of these like weird colors in them, and that's the bacteria that are living inside of them. So these thermophiles are, again, not very relevant to human sickness, to human health, but they can be very environmentally important. Up above that, we get the hyperthermophiles. They like it really hot. They live from 70 degrees Celsius, that's scalding, to 110 degrees Celsius. Boiling is at 100. So this is water that is literally hotter than boiling. You've never seen water that hot because water here boils at 100, right? So unless you're looking inside of a pressure cooker, water doesn't get that hot. The place where you do find water that's that hot is at these like black smoker vents at the bottom of the ocean, and then all of the pressure under the great pressure of the ocean. You can have water that's at 110 degrees Celsius, but you don't find it naturally occurring most other places. These guys are mostly archaea. They're some of those extremophiles that like to live in extreme environments. Practically none of these are human pathogens. Very little of them are going to be relevant to human existence on a daily basis. I mean, they're a part of the general ecology of life, but places where they live tend to be places where where we don't live. They're too wild, even for us. So if we're talking about oxygen, with these pictures, what I want you to think about is this test tube contains water or some other liquid through which oxygen can diffuse. And so at the top of these vials, you're going to get lots of oxygen. And farther down, you're going to get less. And then at the bottom, you're going to get practically none. And so that's what these pictures represent. And you would think, in general, because we require oxygen generally on a daily basis, you sort of think that, well, most things probably require oxygen, or most things, want to have oxygen. It's a good thing, right? Not for everything. In fact, oxygen is a poison. Many, not all, but many of the symptoms that we collectively call aging are in fact caused by oxygen and by the toxins and reactive molecules that are released by the digestion of oxygen. In theory, if you didn't have to breathe oxygen, you could live forever. Of course, I don't know anyone who can hold their breath that long. So we have about 5 categories of things that live at various oxygen levels. First is obligate aerobes. That's here on the left. That's what you are. That's what I am. That's what, most, not all, but most eukaryotes are. Plants, cats, dogs, and some, though not all, bacteria. Obligate aerobe. So an obligation is something that you have to do. And aerobe, you think aerobics. What do you have to do when you're doing aerobics? You got to remember to breathe, right? So an obligate aerobe is something that has to breathe oxygen. You're only going to find them in places where oxygen is common, like at the top of this tube here. You won't find them living very far underground. They're going to be in the very topsoil. You might find them living in, on your skin. Certainly a high oxygen environment. In your lungs can be a high oxygen environment. In your intestines is actually a pretty low oxygen environment. Most of the gases that are in there are produced by the stuff that's growing in there, but there's some oxygen that gets in there sometimes. But things that just live out in the environment might be obligate aerobes, but they don't have to be. There's a few other things that they could be. The next thing, moving towards the right, is facultative anaerobes. This is probably one of the bigger classifications in terms of numbers of bacteria in it. Facultative anaerobes. Facultative, a facility is an ability that you have, okay? But not something that you have to do, just an ability that you have. Anaerobe, and this is the opposite of an aerobe, right? Not using oxygen. So this term means that they have the ability to not use oxygen, but they don't have to not use oxygen. They can use oxygen. They're aerobes. They like oxygen. Oxygen helps them out. They grow better with oxygen. But, unlike you and me, if they don't get oxygen, it's not the end of the world. They slow down. They don't grow quite as well, but they won't die. They can switch to that anaerobic metabolism. A good example of these is yeast, right? So yeast out in the normal world, if there's plenty of oxygen around, they're fine, they're great, they like oxygen, that's cool. But if you restrict the oxygen that they can get, then they'll switch over to an alcohol fermentation metabolism, and then they won't use oxygen when they're making ethanol. It's not as efficient. They don't get as much energy. They don't grow as fast, but they can do it. So in this picture, what you see is at the top, the cells look big and healthy, right? Whereas at the bottom, the cells look smaller. So that means that the cells are healthier, they're growing faster at the top where there's lots of oxygen. At the bottom where there's not any oxygen, they're still alive, but they're growing slower, they're smaller, they're less healthy cells. Moving over, you have obligate anaerobes. This is just the opposite of an obligate aerobe. They can't have oxygen. Oxygen is a poison to them, it kills them. Like you and me and other aerobic things, even though oxygen's still a poison to us, we have defense mechanisms against it. We are chock full of stuff that helps us deal with the fact that we're living in oxygen. These guys, obligate anaerobes, they don't have those, right? Oxygen has its full potent poison effect on them and just kills them off like nothing. So at the top of this vial, you're not going to see any of them. The oxygen kills them off. They can only live at the very bottom where there's no oxygen. Right, next one, microaerophiles. These I call the Goldilocks bacteria. You know, Goldilocks, this porridge is too cold, this porridge is too hot, this porridge is just right. That's microaerophiles. And they sort of get the shaft from both ends, right? They're aerobes. They're obligate aerobes. They have to have oxygen. But they don't have those so much of those special protective things. Like, for instance, a big one is catalase, which is an enzyme that helps degrade oxygen byproducts. Like, they don't have as many of those. So oxygen, even though they have to have it, it kills them. So they can't live at the top. There's too much oxygen there. It just wipes them out. They can't live at the bottom because there's not enough oxygen. And they have to have oxygen to live. So they only really live in the middle, where there's just enough, but not too much oxygen. A lot of the bacteria that live in your mouth are microaerophiles, and they'll actually, like, not the ones that live just on the surface of your mouth, but like the ones that get buried down deep, like, you know, in the gunk between your teeth. That's a nice place where they can get some oxygen, but not too much of it. So some of those bacteria in your mouth are microaerophiles. Some of them are obligate aerobes, and a lot of them are facultative anaerobes as well. Not a whole lot of obligate anaerobes are human pathogens, but there are a few. The last category here is aerotolerant. And a lot of people get these confused with facultative anaerobes, but they're very different. Aerotolerant are anaerobic. They do not use oxygen, but they do have the protective elements that keep them safe from oxygen. So that's why they're aerotolerant. They don't use oxygen personally. Like, they go, oh, oxygen, I don't use it myself, but I don't have a problem with oxygen. They're very tolerant of oxygen, so they can live anywhere. They can live at the top of the vial. There's oxygen there. They don't care. They're not using the oxygen, but it doesn't hurt them. They can live at the bottom of the vial. There's no oxygen. That's fine, too. They didn't really need it anyways. They differ from, even though both them and facultative anaerobes are going to live all throughout at any oxygen concentration, facultative anaerobes are going to do better with oxygen, and they're going to grow slower and less healthy without oxygen. They can still grow, but they're not as cool if they don't have oxygen. Aerotolerant, they're the same no matter what. Oxygen, no oxygen, they don't care. Alright, so if we're talking about pH, there's basically 3 different categories, and they're pretty simple. Neutrophiles, acidophiles, and alkophiles. Vast majority of bacteria are neutrophiles. Pretty much all, well, most human pathogens are neutrophiles because the human body is a neutral environment. So most things that are going to live in us will want to live in neutral environments. There are acidophiles. They live in acidic things. As far as humans go, there are a lot of, I would say, food production important bacterias that fall into this category. There are the lactic acid bacterias that take lactose in milk and turn it into an acid. These are useful in making cheese and yogurt. They're also useful in, well, not useful, but there's something you have to watch out for in just milk that spoils. There are the bacteria that take, like the alcohol in wine and turn it into acetic acid to make vinegar. Vinegar is something that's made by microbes, and this is something that those microbes do because they want to live in an acidic environment. They love being in an acidic environment. Most other bacteria hate it. So by making their environment more acidic, they can feel more comfortable, and simultaneously they can kick out all of the unpleasant people that they don't like and monopolize this nice little vinegar stuff to themselves. So as far as humans are concerned, that's where you're going to find most acidophiles. Alkophiles grow in basic environments, so with a high pH. There's not a whole lot of alkophiles which are super relevant to humans from a health perspective. From an environmental perspective, there's a lot of soil can be alkaline, and you're going to have alkophilic bacteria that live in there. They can be very important for things like agriculture or ecology. But from a health perspective, not a lot of them are pathogens. The last thing to keep in mind is water availability. And in this case, we're not necessarily talking about like how dry something is. That's a part of it. But water you can be surrounded by water and still die of dehydration. Like, you may have heard that if you're stuck or stranded on a desert island, you shouldn't drink the seawater because it will actually dehydrate you. That's true. If you drink salt water, you will lose more water than you get in. You could be drinking all day long and you'll die of thirst. It's kind of crazy like that. And it's the same with microbes. If they're in salt water, then osmosis from the environment, or then osmosis from the salt that's around them is actually going to draw water outside of the cell and into the salt water around it. And most bacteria do not like to go into high salt or high sugar or high solute levels for that reason. They'll lose their water and they'll die. This is why we pickle things in briny salt water to preserve them. Even though they're sitting there in water, it's water with enough salt in it that most microbes don't like living there. It's the same with preserves, like jam. We're using sugar instead of salt, but still it's got so much sugar in there that the sugar is going to pull the water out of anything that's trying to live in there. Same with honey. You may have heard that honey is a natural disinfectant. It does have some natural disinfectant properties, but most of the preservation property of honey is just the simple fact that it has so much sugar in it that it will just steal the water right out of anything trying to live in it. So nothing really does, or very few things. There are some organisms that can grow in these like high salt, not usually high sugar, but usually it's a high salt environment because that's what you find naturally. You only really find high sugar environments in things that humans or sometimes bees have made. You find high salt environments all around. You got the ocean, you got the salt sea, you got the salt lake, that sort of thing. And there's two classifications of these type of organisms. Osmotolerant, just like we said, the aerotolerant, like they don't need oxygen, but they can live with it just fine. That's not a problem. Osmotolerant don't need high salt environments, but if they find themselves in a high salt environment, they're cool with that. It isn't going to hurt them. They go, oh, isn't there a lot of salt around here? That's very interesting. And it doesn't affect them one way or the other. The other type are halophiles. So halo means salt, phile means to love. They don't just tolerate the salt, they love it. They've got to have it. And they usually have specially designed water pumps that keep pumping the water back into the cell. And if they get into an environment that doesn't have a bunch of salt in it, then they just continue pumping the water in until they explode. So they not only can live in these high salt environments, they thrive in them. And if you try to move them to a not high salt environment, they usually die. So, those are the main environmental or physical factors that influence microbial growth. And in my next installment, I'm going to talk a bit about thermodynamics and energy and metabolism, and how energy and metabolism work specifically in microbes.